Hi everyone, I'm Dr. Sister Arul Sheeba Rani, working as Assistant Professor, Department of Botany, Nirmala College for Women, Coimbatore. I welcome you all for this learning module. Now I am going to explain about the structure, types and development of endosperm. Endosperm, the primary endosperm nucleus divides immediately after fertilization but before the zygote starts to divide into an endosperm. The primary endosperm nucleus is the result of triple fusion, that is, two polar nuclei and one sperm nucleus fused together and thus has 3n number of chromosomes. Endosperm is a nutritive tissue and regulatory structure which gives nourishment to the developing embryo. Depending upon the mode of development, three types of endosperm are recognized in angiosperms. One is nuclear endosperm, second is cellular endosperm, and third is helobial endosperm. This is the structure shows the types of endosperm, how it gets developed. Next is nuclear endosperm. In the nuclear type, the primary endosperm nucleus divides into two without any wall formation. The subsequent division of these two nuclei are free nuclear, so that the endosperm consists of only free nuclei and cytoplasm around them. The nuclei may either remain free or may become separate by walls in later stages. Examples are coccinea, capsella and arachis. This is the structure shows the coccinea and the peanut seed. And this is the stages of development of nuclear type of endosperm. Next is cellular endosperm. In this type, the primary endosperm nucleus divides into two nuclei which are immediately followed by a wall formation. Subsequent divisions are also followed by walls. Examples are Adoxa, Helianthus and Scoparia dulcis. This is the structure shown for the Scoparia dulcis, and this is the cellular endosperm developmental structure. Next is helobial endosperm. In helobial type, the primary endosperm nucleus moves towards the base of the embryo sac where it divides into two nuclei. These two nuclei are separated by a wall to form a large micropylar chamber and a small chalazial chamber. The nucleus of the micropylar chamber undergoes several free nuclear divisions, whereas that of the chalazial chamber may or may not divide. Examples are hydrilla and vallisneria. This figure shows the developmental of helobial type of endosperm in Erimurus. And this is the image of hydrilla. The endosperms may either be completely consumed by the developing embryo or it may persist in the mature seeds. Those seeds without endosperms are called non-endospermous or ex-albuminous seeds. Examples are pea, groundnut and beans. Those seeds with endosperms are called endospermous or albuminous seeds. The endosperms in these seeds supply nutrition to the embryo during seed germination. Examples are paddy, coconut and castor. Ruminate endosperm. The endosperm with irregularity and unevenness in its surface forms ruminate endosperm. Example is Areca catechu. The activity of the seed coat or the endosperm itself results in this type of endosperm. The unequal radial elongation of the layer of seed coat results in the rumination of endosperm in Passiflora. In Anonaceae and Aristolochiaceae, definite ingrowth or infolding of the seed coat produces ruminate endosperm. This is the structure of ruminate endosperm. This is the cross section and longitudinal section. Endosperm hastoria. Another interesting feature of the endosperm in its presence of hastoria. In the case of helobial endosperm, the chalazial chamber itself acts as a hastorial structure. In cellular and nuclear endosperm, special structures are produced towards the micropylar chalacel, both micropylar and chalacel, which may be in lateral direction depending on the species. These absorb nutrients from other outer tissue or from ovary tissue and supply them to the growing embryo. The irregular surface of the seed coat makes endosperm ruminate in myristica fragrans. This is the structure shows the developmental types of seed, outer integument, and this is a proembryo inner integument, and these two are lateral endosperm hastoria and inside the nucellus area. And this is the developmental structure of hastoria. And there are functions of endosperm. It is the nutritive tissue for the developing embryo. In majority of angiosperms, the zygote divides only after the development of endosperm. Endosperm regulates the precise mode of embryo development.